Good. So without further ado, we're going to plough on into the main part of the agenda. Gives me enormous pleasure to welcome Ken Smith. Ken's laboratory here in Cambridge has really been the driving force for the development of this biomarker. We're going to be hearing a bit from Owen shortly as well, and from James as well. These have been the guys who are the scientific brains behind the development of this biomarker. So Ken, we look forward to hearing your talk about translation of biomedical advances. Ken Smith, I should say, is our Professor of Medicine here in Cambridge and runs as a nephrologist, although we've gradually lured him towards the world of inflammatory bowel disease, I'm delighted to say. And Ken now has a big interest in IBD, which is terrific. Thank you, Ken.
Thanks, Miles. Dragged rather than lured, perhaps. So thank you all for coming. I am a nephrologist, so there are things I'm going to tell you that I clearly don't know anything about, so please ignore those. And I'm looking forward to the day, actually. It's going to be a lot of fun. I was pretty pessimistic actually about the walk from here into town, because I know if a lot of nephrologists did that after a long day past the pubs that Cambridge has, not many would arrive at their destination. Miles tells me that you're all completely different. So what I'd like to do is to find out how to start the talk. Yeah, thank you. Is to provide some background into where all this started, and also to address the issue more generally of prognosis in inflammatory disease. So prognosis is something that actually a lot of us don't think about that much. Has anyone seen this film, by the way? That's reassuring, because it was never made. This is a this is a film poster that apparently appears on the wall if you watch episodes of Friends. If it was made, it would probably be the only ever film talking about prognosis. So in my lab over many years, we've been interested in a number of autoimmune and inflammatory diseases. So some of them are listed up here. We started off predominantly with an interest in ANCA-associated vasculitis and lupus, and have moved after James Lee's arrival in the lab to also work on inflammatory bowel disease. But we have interest in various other diseases. And one of the things that is common to all of these diseases is they have very different prognoses. So if you have lupus or vasculitis or multiple sclerosis, we all know that many people with those diseases do very well in the long term and have pretty normal lives, even if they're not cured, and respond to sort of therapy. But we also know there are subsets of people who do badly and who don't respond to therapy, who have ongoing, remitting, relapsing or persistent disease, and they have shortened lifespans and chronic disability. So something actually I quite like doing, and I will do with you, is to suggest to you that you have to choose one of these diseases to have when you leave here. So if you had to choose one, and we'll give you a choice, we won't give you IBD because you know too much about that. If you had to choose between having lupus or multiple sclerosis, one or the other, who would have lupus? Okay, that's about a third of you. Who would have multiple sclerosis? You've got to choose one or the other. This doesn't bring bad luck. So most of you haven't answered the question, and that's because it's a relatively stupid question. And the reason it's a stupid question is that actually I don't care which of these diseases I have, as long as I'm in the group that does well in the long term and not in the group that does badly. So from a patient's point of view, actually prognosis is more important, I think, than diagnosis. And we're very good at putting people into these different categories, but we're very bad when they turn up to the doctor at working out what their long-term future holds. Are they going to have quiescent disease or relapsing or persistent disease? So to try and address the pathways that drive these different outcomes and see if we can predict these different outcomes, we set up a program in the lab a long time ago, and this was initially set up by Paul Lyons, who's up the back there, staring at his computer because he's heard this before, and he's an immunogeneticist who's worked with me for a very long time. So we recruited our first patient into this study in 2004. And what we decided to do was to recruit patients with autoimmune or inflammatory disease. But what made it different to a lot of other studies was we recruited them at the day of diagnosis. So they hadn't had previous therapy. This was the day they turned up and their diagnosis was made. And on that day, we took blood, about 100 mils of blood, and separated these 5-cell subsets, neutrophils, monocytes, CD4 and CD8 T cells and B cells. So we purified those, and we did transcriptional analysis, whole genome transcriptional analysis of each of those subsets individually. And our experience was that that allowed us to discover a lot of interesting things, none of which were visible if we just looked at whole blood. So the separation of cells is critical. And the third thing we did was to follow these patients up in the long-term detailed clinical phenotyping. So these are all single-centre cohorts. We now have follow-up, for example, in our lupus and vasculitis patients of a median of almost a decade on our first cohorts. And that allowed us to look back from a vantage point of years into the disease to see what was going on at the start that could predict what subsequently happened. So when Owen McKinney, who's also here somewhere and will be talking next, came into the lab to do his PhD, we gave him this giant data set and said make sense of it, and he went about and spent a few years and did that. And so the first thing he found that was interesting was that in CD8 T cells, purified CD8 T cells, there was a pattern of gene expression that divided patients into two very distinct groups. A group here, this red group where all of these genes are up and these are down, and a blue group where the reverse was true. And in a second cohort, and these are patients with ANCA-associated vasculitis, in a second cohort he found the same thing. So the genes that cluster those two groups can recluster the first cohort and vice versa. So this was a reproducible finding that patients fell into these two very distinct groups. We then spent a lot of time trying to work out what this meant, because no matter how hard we looked, we couldn't see any clear difference at presentation between these two groups. They had the same level of organ involvement, the same sort of comparable disease activity, same genders, and in fact the same response to initial induction therapy. But what was different, and we did this on a number of different platforms, and this is not a batch effect. What was different about them was what happened next. So even though they were similar at the start, the patients in the red group, and this is time to first relapse in vasculitis, about 80% of them had relapsed by 3 years, whereas the patient in the blue group, a few relapses early, but did very well after that. And this separation holds true out now to 8 years. So these people have very different long-term disease behaviour, but diagnostically at the start were the same. And this is the course over time. Each red dot is a flare of disease, and you had to obviously have active disease to get in, so they all have a red dot at the start. But the patients in the red group usually had multiple flares over time, and the patients in the blue group, apart from this one, had either no flares or a single flare. So they're very different long-term disease behaviour. Owen did the same thing in our cohort of lupus patients. He found a very similar and statistically the same set of genes that divided patients into two groups of similar relative sizes, and that too predicted their long-term outcome. And then James Lee came into the lab, and James gathered cohorts of patients with ulcerative colitis and Crohn's disease, and this is time to escalation in therapy in those two diseases, and he found the same thing. Same sort of division, similar or overlapping gene sets involved, and a profound long-term prediction of outcome in these two cohorts. So this observation held true even beyond the diseases I've shown you. So using this prognostic signature or a surrogate, Owen's managed to demonstrate that this can predict good outcome in infection, HIV or Hep C, and vaccination to these three vaccines, while at the same time predicting poor outcome in autoimmune diseases. So I've mentioned vasculitis, lupus and IBD, but this seems to hold true in very early type 1 diabetes, idiopathic pulmonary fibrosis and others. So these are common pathways that work across different diseases to drive outcome. A couple of interesting asides. When we look for this signature and this separation in patients who don't have active disease, we don't see it. There's no clear separation, and if we did a blood test of all of you now, provided you're well, we wouldn't see separation into two groups. But if we vaccinate you, 3 days later you will be separable into two groups, and that response will predict which group you go in will to an extent predict your vaccine response. If we vaccinate these patients while they're in remission, we can induce the signature and they go back into their initial group. In other words, this seems consistent over time, whether you're in the red or blue group, in a given individual, and it seems independent of the antigen we give. The disease initiates the same sort of signature as a flu vaccine would, for example. So I'm going to not talk more about the signature because both Owen and James will address very relevant aspects of it in the next two talks. I want to go on to a quite different approach to thinking about prognosis, a genetic approach, which allows, I think, us to make some general points about the prediction of outcome in autoimmune diseases and in particular, and inflammatory diseases, and in particular IBD. So the question we then asked, this has now got nothing to do with transcription, it's got to do with genetics. Do common genetic variants control the outcome independent of diagnosis? And this picture, which was one of our students, John Sowerby's attempt to get on the cover of Cell, which failed, but will be the meaning of this with this fox here and his disease burden will become apparent later. So this was a study put together by James in collaboration with Miles and the IBD Genetic Consortium, among others. And essentially it's a Crohn's disease candidate gene analysis, but not by diagnosis, but by outcome. So the IBD Genetics Consortium, like all genetics consortia, had compared people with disease to people without disease and looked at the genetic variants that differed between the two to give an indication of what genetic variants predisposed to getting disease versus not getting it. So James took a different approach. He got a bunch of patients, 1,800 patients with Crohn's disease, and within them found a subset who he called flarers, who had severe disease, had 2 or more immunomodulators or surgery or at least one of each, or fizzlers who'd been followed up for at least 4 years and had had none of those things. So in other words, the end of the spectrum who'd done pretty well and the end of the spectrum who hadn't done so well, and compared the genetics of those two. And initially compared this using just 81 genes curated on the basis of the red signature I showed you right at the start, because we didn't think we had the statistical power with these sorts of numbers to look at the whole genome, because using fewer genes allows us to have a lower level of statistical significance to meet. And I'm going to summarize about half his PhD in this single slide. I sort of like doing this, but I can see why he may not like me doing this. So what he found was a variant in FOXO3A, which is a transcription factor, the G allele, was associated with a mild course of disease. And it was associated with that disease in 4 independent cohorts and reached genome-wide significance. You might argue, why did you need to reach genome-wide significance? Because you set this up with 81 genes in order not to have to reach genome-wide significance. And we argued that with the editors for a long time, and they said that they agreed with us eventually that there was no statistical requirement for genome-wide significance, but there was an editorial requirement. So we were forced to go out and James was forced to go out with collaborators, some of whom are here, and get 2 more cohorts from our initial 2 to make 4, and that got us to genome-wide significance. So we're confident this is real. And the interesting point, which I'll come back to, is that while this was associated with outcome, it was not associated with Crohn's disease susceptibility in the much larger IBD susceptibility cohorts. And what James went on to show was that this G allele was associated with increased FOXO3 transcription and recovery of nuclear FOXO3. That bounded directly to TGF-beta promoter, that increased TGF-beta, and TGF-beta then reduced all of these pro-inflammatory cytokines and increased IL-10, so this is in vitro. Now, and this is in a monocyte-driven pathway. Now, there are other potential mechanisms by which this might work, but the fact that it's TGF-beta dependent and has this profound effect on all these different pro-inflammatory cytokines make us wonder if maybe this is actually not just restricted to Crohn's disease, maybe this works across different diseases. And we found that the same association with good outcome was found in rheumatoid arthritis, if you measure rheumatoid arthritis progression from onset, and the opposite was found in malaria. So the same value was associated with severity in malaria, both in Vietnam and in Kenya, so in 2 different populations. So again, this seems to be a common pathway driving outcome in a range of diseases. So what we then decided to do was to scale this up so we could reach genome-wide significance more confidently in now 2,600 patients with a slightly larger flare and fizzler cohorts. And so this is a standard genome-wide association study, I should say. I'm not going to go into the details. Daniele Biaschi was a key person involved in analysing the data. But we had the primary cohort, a replication cohort, and we used a standard biobank array, a very good array actually, and GWAS analysis. And what we found were there were 4 variants that reached genome-wide significance, shown here. So these are variants that associate with outcome in Crohn's disease. So they're shown here, exact on the X chromosome, an MHC, which I'll come back to, association, fortunately and reassuringly, FOXO3 and IGF-BP1/3. These are the sort of loci that were implicated. To demonstrate that this wasn't just some sort of artifact of looking at the extremes of the population, James went and looked back at those 2,600 patients and divided them into 4 different subgroups based on a gradation of prognosis from severe to mild or mild to severe, and then looked at the association between the genetic risk score based on these 4 variants that I've talked to you about, sort of summed, if you like. And you can see that titrates across prognosis, right across the population. So this is not an artifact of looking at extremes. This is something that associates with everybody's graded prognosis. And similarly, an alternative definition of severity or poor outcome in Crohn's disease is the requirement for surgery within 2 years of diagnosis. And again, that's strongly correlated with these genetics. So how am I going for time?
You've got about another 3 minutes.
3 minutes. So I'm going to duck over some of the detail here because it is a bit nerdy and get on to the, sort of fun though, get on to the key point. So the key point is that none of these prognosis-associated variants were associated with susceptibility to Crohn's disease, even using the IBD Genetics Consortium meta-analysis, which at that stage had about 6,000 cases and 15,000 controls. And conversely, none of these susceptibility associated variants were associated with Crohn's disease prognosis. So none of the 170 alleles, even if we did a liberal Bonferroni correction, but importantly after stratifying for disease location. And you can see that these genetic risk scores based on these 170 susceptibility associated alleles don't associate with prognosis, and we can do this in different ways and get the same result. So that's the key point, that a genome-wide analysis of prognosis in Crohn's disease finds variants that are strongly associated with outcome. All of them are plausible candidates. That's the bit I didn't show you. But they don't associate with susceptibility, and susceptibility doesn't associate with prognosis. So genetically controlled pathways driving outcome appear to do so at least largely independent of susceptibility. Now, if you're an immunologist, that makes sense. If you're dealing with an autoimmune disease, for example, this isn't, I know, that requires a break in tolerance, why should the factors that drive that break in tolerance remain relevant once you've got an ongoing inflammatory disease and tolerance is long since broken? Conversely, or similarly, if this is a barrier dysfunction disease, once the barrier is breached and the inflammatory response has started, why do the factors that predispose to that initial barrier breach remain relevant? And we would argue they probably don't. So in summary then, distinct pathways govern susceptibility and outcome. The prognostic signature, which is the main reason we're here today, works across different diseases, predicts outcome, is not associated with disease activity, and doesn't seem associated with susceptibility in that the frequency of that red group is the same whether you've got disease or haven't. We've got a similar story in multiple sclerosis I haven't talked about that says exactly the same thing but is related to a different pathway, and I've just mentioned the genetics. Is that important? Well, we think it is important, and it's important for two reasons. The first reason is that it might give us the ability to predict outcome, and that's what you're going to hear more of in the future, and that could be of benefit to patients. And the second thing is that it could make us think about different therapeutic targets in the long term. Because once you turn up to see your doctor, susceptibility is finished. And what is in front of you is prognosis. And what we want to then do, given that it's too late to prevent disease, is control your outcome and improve your outcome. And it could be that these variants that associate with prognosis are more important than the ones that are irrelevant by the time you turn up to the doctor. So this has been, you'll hear more from these guys, but there's been key individuals involved in driving this project forward over the years, and I've only listed some of them here. But the point of this actually is this has been a remarkably effective collaboration between the lab and the clinic. And that's what's made it work, both with the vasculitis and lupus team for the initial studies and then the gastro team, and our collaborators involved in pulling together these cohorts. And not just pulling together the cohorts, but the very high quality clinical information that you need to make these studies possible. This is not just a genetic study, this is has to be a collaboration with the clinic. Lots of people have funded our work eventually. At the start they didn't because they said it was just a fishing expedition. We pointed out subtly that if you sit at home and don't go fishing, you don't catch fish. That didn't work, but eventually it did, but only once we got the data in. So what you're going to hear from the person next is Owen McKinney, who's going to talk about the mechanism underlying the prognostic signature. Then James Lee about how that was turned into practical tests for IBD and then validated. And finally, delivering this test to the patients is now being done by a spin-out company called PredictImmune, who are here, and you can go and see what they're doing later on today, because of course this is something we can deliver to the clinic in advance of a trial result because it's been validated, as James will point out. So that's what the rest of the day holds. And that's what we hope our future isn't in Cambridge with Brexit.
Thank you very much, Ken.
Thanks, Mark.